Good evening, gang. Another Speaker's Corner. We have a Renaissance man talking to us tonight. Frank is so, what do I say? He has so many interests and abilities, and he's always glad to share them, which is unfortunate for him that I know that, because I pick on him a lot. I've drug him forward 2 or 3 weeks from his intended time, but that's okay. We need to know this stuff now. And I'm not going to talk too long because it's Frank's turn. So let's welcome Frank to talk about everything we never wanted to know about flu, but we get to know.
All right, so we're not going to talk about the symptoms of the flu. You already know those. The chills, the body aches, the fever. That's not fun to talk about. We're going to talk about the biology of the virus. What makes it special? How does it work? And so the influenza virus is an enveloped, negative sense, single-stranded RNA virus. That should explain it all, right?
Absolutely. Go home now.
Exactly. The talk's over, you've got the whole thing. Now, I'm going to go through this and break it down because a lot of these are going to be important for how to understand what kills the virus, what prevents the spread of the virus, how the virus replicates, right? So first, enveloped. That basically means that it's surrounded by a membrane. There's 2 kinds of virus. One has a protein coat, one has a membrane around them. This is an envelope, the membrane-bound virus, right? Negative sense. Now, what negative sense means is that this is a non-coding RNA. So you guys have probably all heard way back when you took biology, the idea that DNA is transcribed into RNA, it gets translated into proteins, right? Well, the coding strand, right, the coding RNA is what goes into proteins. The other strand is the non-coding. So this viral genome, the influenza virus, doesn't actually make the protein directly. It has to get made into a coding strand of RNA, right? So that's a big difference. And it has to provide an enzyme to do that because your body goes DNA to RNA to protein. Simple. Well, the virus changes that a little bit and lets it go from RNA to a coding strand of RNA, and then that gets made into proteins, right? And we'll talk about how that happens, but it uses its own enzyme, an RNA-dependent RNA polymerase. And this is one of the special viral enzymes that it makes. This virus only has 17 genes in it, right? To give you an example, humans have about 26,500 genes. E. coli has over 4,400. This virus has 17. Very small, relatively simple, and it's going to steal functions from the cell. So it's going to do everything that the cell usually does, but for its own purposes. It's going to basically take over the cellular machinery and make more copies of itself. All right? So, single strand. You guys have all heard of the double-stranded RNA molecule, right? Well, double-stranded DNA. RNA is single-stranded. And so it basically deoxyribonucleic acid and ribonucleic acid are very similar to each other. There's just that deoxy. It's missing a particular group called a hydroxyl group. Well, if you're missing that, it can form these nice long double-stranded helixes that can go on for 1 million bases or more. Okay? RNA doesn't do that. It can make these short little separate strands of DNA, of double-stranded material, so it basically folds back on itself and makes a little knot. And so these RNA molecules actually have a structure, but it doesn't have a nice simple structure like DNA. Okay? And that's going to be important when we're talking about the packaging of these RNAs into the virus. Never thought you'd all of this in a talk, right? Yeah, this is more biochemistry than probably you've had since high school. Right. Like I said, this is a lot of these things you've heard of, but I'm going to try to explain how these things come together and make a virus that's actually more dangerous than people assume, right? People don't worry about the cold. You get colds, there's over a couple hundred viruses that will cause symptoms like a cold, which are very similar to the flu, but everybody talks about how dangerous the flu is. They have a flu vaccine, all these other things happening. Why is this special? Well, we're going to talk about what makes influenza so much more dangerous than these other cold viruses, and it all comes down to how the virus behaves and what it can infect, right? So, these flu viruses are not new, right? Well, you guys have probably heard of Hippocrates, the Hippocratic Oath, way back in ancient Greece. He was the first one to describe the symptoms of the flu that people could recognize, right? And, you know, they're the same ones that we have now. We worry about influenza because it causes so much disease. Most people don't realize just how many people get killed annually. So typically, in a regular flu season, roughly 300 to 700,000 people a year around the world die from the flu, right? To give you an idea, 415,000 people died of COVID in 2021, right? So you're talking American COVID levels every year around the world, right? The US, we don't have that many flu deaths average. We get 20, 30, 40,000 a year dying from the flu in most years, right? Except for 2021. Gee, I wonder what's happening around 2021 that might have stopped the spread of a respiratory airborne virus. You know? Yeah, we know how to slow down the spread of flu. Are we willing to pay that price? Right? The methods work, they're just costly in terms of people. Right? And typically, you have roughly 10 times more people who go to the hospital with the flu than actually die of it. Right? So this has a very heavy healthcare burden. And what makes it even worse, this is just a typical year. Every now and then you get a bad year where you have these epidemics, right? And they happen pretty routinely. One of the first ones was way back in 1510 called the African flu. The Mediterranean suffered for that one for many years. 1580, whole towns were getting wiped out, but they didn't keep very good records because, well, the town got wiped out. Right? 1680 to 1699, when people were settling in America, coming over from Europe, they took the flu with them. And they had these repeated epidemics in different places around the globe because of it. 1890, they caused more deaths than cholera, which was very common at the time. Right? Then you get to the really big one. 1918, 1919, the Spanish flu. Right? We hear about it called the Spanish flu, and the only reason it's called that is because they were the first one to record it. They reported it because they were neutral in World War I. All these other countries didn't want to admit how badly they were being affected by this flu. Spain was willing to talk, and partly because people weren't able to control the spread very well, they weren't sharing information. Somewhere between 30 and 50 million people died. Remember, this is when you had a much smaller population, and so the population then was about 1/5 what it is now, run about 1.5 to 1.8 million people. This particular virus caused more deaths than World War I did to combat. Right? Given a better idea of how many people were infected, typically around 7% of the population gets the flu in some kind of form, not necessarily hospitalized from it. Well, this year about 1/3 of the population got the flu. This was a very dangerous one. But, you know, it wasn't the only one. 1957, the Asian flu killed about 2 million people worldwide. 68, the Hong Kong flu, 3 million people. 2009, the swine flu, about 1.3 million people. Right? Influenza kills. And we tolerate it as a disease that pretty much everybody gets, but it's there, and it's not going away, right? So, why is this so dangerous? What makes influenza such a nasty problem for the world? Well, basically it comes down to a number of issues. First off, RNA. It's an RNA-based virus, and RNA is a lot less stable than DNA. It changes more quickly, around 1,000-fold more quickly, right? So it can have a mutation, and if you're making 300-400 viruses per cell, one dead virus, no big deal. All the others are going to go around infecting more people, and because it allows all of these changes, if any of these strains become different and become better at infecting somebody, then you get more of it, and it gets to spread more, right? This is known as antigenic drift, and this drift means a slow change over many generations, right? And that's not the worst of it, right? The bigger problem is actually how many different kinds of animals influenza is able to infect, right? Different strains of influenza can infect roosters or chickens, humans, bats, pigs, horses, dogs, cows, whole bunch of different animals. And that's a problem, because when you have two different strains of influenza coming together in a particular animal, they can get mixed up. And you can have part human, part fruit bat virus that we don't have any immunity to, right? This is where they think a lot of the pandemics come from. This is a process known as zoonosis, where basically the virus goes from infecting one species to another species, and this is known as an antigenic shift. Drift is when it mutates and changes a little bit and evades your immune system. Antigenic shift, much bigger changes, typically means a much bigger flu season because of that, because your body hasn't seen that. Okay? And so you have these shifts, these part of one virus joining another part of a different virus. This can happen routinely, it happens frequently. Many of them don't work, right? If you have a bad recombination, the virus is dead, no big deal, right? There are still 300 more from that same cell that are going out and infecting people. And the last reason is one you probably all experienced. Have you ever had that cold or flu that just wouldn't go away? You know, you get sick in December, you start feeling better, you get sick again in January, maybe you get bronchitis that lasts until March, you start feeling better, and you get another flu. This process of just constantly fighting off infections really weakens your body. And when it's weaker, other diseases come in and take advantage of that weakness. And the flu can be pretty debilitating, especially if it keeps changing a little bit and reinfecting you. Right? So these are some of the reasons that influenza is actually a lot more dangerous than the common cold, right? So, how does influenza infect a cell? There are two main proteins involved in this process, right? There's 17 genes, but two main functions are going to be needed for these cells to get infected. And you actually probably heard of these, even though you may not have realized it. You have hemagglutinin, or H, and neuraminidase, or N. The H1N1 flu, named after the type of hemagglutinin and neuraminidase that you find on that virus particle, right? This is one of the most common forms. H3N2 is another one that was in this year's flu vaccine. H5N1, this is the swine flu that's actually spreading around in birds and cattle right now, right? These are named after the two main proteins that are going to determine what can get infected and what won't be. So, hemagglutinin, 18 different types, 3 main ones infect humans, plus this new H5 one that has so many people worried, right? Different hemagglutinins are going to be used to infect different species, So you can mix and match these viruses later on. And it has a particular function to bind a 9-carbon molecule known as sialic acid. Right? Now, the interesting thing is neuraminidase cleaves sialic acids. It cuts it off of cells. Right? We're going to talk about why these are different, but basically, hemagglutinin is used to get into the cell. Neuraminidase is used to get away from the cell. You have to be able to get it off the cell so that it can go infect other cells. Okay? And basically, these are found in a number of different viruses. Hemagglutinins, they're involved in things like measles, polio. You might have heard of these diseases, right? They also use this hemagglutinin to help get into other cells. Okay? Now, it's a common protein in viruses. It'd be really, really nice if we could come up with a way of blocking hemagglutinin function. But as I'm going to show you in a little bit, we're not quite sure how it works, so it's hard to block something you don't understand. Neuraminidase, on the other hand, we have a good idea of what it does, and we'll look at that more closely too. Right? Any questions on the idea that you have these two main molecules? This is what the viruses are named after, and they are going to do different things? Yeah, Randy.
So there's 18 H's and 11 N's. Does that mean there's about 198 kinds of flu?
So the question was 18 kinds of hemagglutinin, 9 kinds of neuraminidase, maybe 200-ish kinds of flu. If that were the only thing that were different, then yes, but basically these hemagglutinin and neuraminases, they're used to determine what species you can infect, right? Haven't mentioned H4. H4 doesn't typically infect humans until you mix it maybe with a little bit of H1, and now we have a new hemagglutinin molecule, right? They have a very broad species distribution, and most of these forms don't infect humans. It's really hard to eradicate something that's living in 100 different kinds of species, right? We're not getting rid of the flu anytime soon. Okay. Other questions? All right, so I'm going to talk more about how these molecules are involved in the process of infection and how the virus actually gets into the cell and what it does. Right? So basically, you have the virus, it binds to these sialic acids on the surface of the cell, and the cell says, okay, I've got a foreign body. I'm going to bring it into the cell and break it down. Right? You guys have all heard of a lysosome, right? One of those favorite little cellular organelles that people forget about as soon as they graduate from high school. Right? Basically, this is a digestive organ in the cell. So what happens is the virus gets brought into the cell. It's called an early endosome, and then the cell basically pumps hydrogen ions in. It makes it acidic. How many of you have ever pickled vegetables or pickled cucumbers or something like that? Right? Same process. You use something like acetic acid in vinegar or citric acid. You put this in, and some of the proteins break down because of these acidic conditions. Your stomach is very acidic. It's used to break down these foods. The cells, or the stomach, learn from the cells, right? The cells start acidifying this compartment. It's supposed to break down the protein, but the problem is this virus particle doesn't want to get broken down and instead fuses with the edge of the lysosome, the edge of the endosome, releasing what was inside of the cell, inside of the virus into the cell. Right? All those RNA particles that used to be trapped within the virus now escape into the cell, where they can go into the nucleus, make their sense strand of RNA, and that sense strand goes out and makes your protein to make more virus particles. Right? It takes advantage of what the cell normally does to break down the virus in order to escape from it. And that's the job of hemagglutinin. It's going to be responsible for letting that virus into the cell body and actually getting in where it can infect things. Right? So now I'm going to show you a model here. Right? This is an atomic scale model of the hemagglutinin protein. And I was trying to come up with a good way of describing how small this is. Right? And the best thing I could come up with was to compare it to things on the Odyssey and our life. Right? So this protein molecule is about 3 nanometers across, 30 angstroms, 10 to the minus 9th meters. What does 10 to the minus 9th mean? Well, 10 to the 9th, 1,000 times 1,000 times 1,000. How big is that? Any of you go to Ponta Tomba to see the penguins while we were here? Right? It's about 200 kilometers away. So let's cut it down by 1,000. 200 kilometers goes to 200 meters. 200 meters, the length of the Odyssey. That's 1,000. Right? 200 meters, cut down another 1,000. 200 millimeters. Maybe, this big. The size of one of the small dinner plates in the cafeteria. That's another 1,000. Let's cut it down by another 1,000. 0.2 millimeters. Anybody ever use one of those really fine pens that draws a little tiny line that often jams up in that? Those are about 2.2 millimeters. One of those lines, compared to the distance to Ponto Tombo, is about the difference between a meter and the diameter of this molecule. I say small, I'm not joking. This is a tiny little molecule. Another way to think about it, this width right here is about the same width as you find on some of the most modern microprocessors, about 3 nanometers. That 3 nanometer process they always advertise. Little tiny things. Right? And this is the molecule that's going to help the cells infect or get infected by the virus. Right? And so you can always play with these things. You can move it around. And I want to point out just a little tiny part of this molecule. These things are known as alpha helices, and believe it or not, these things play a critical role in getting the viral membrane to fuse with that of the endosome. Right? This is what actually releases the virus. Right? The way it does this, right? Basically, under normal conditions outside the cell, you have hemagglutinin, and it binds to these sialic acids. Right? It holds the virus membrane right near the cell membrane. The cell starts acidifying this. Okay? The pH goes down. It becomes more acidic, just like you would when you add vinegar to cabbage or whatever you're trying to pickle. Proteins start to separate. They start to break down. This part of the protein comes off of the center. And remember that little tiny alpha helix I was pointing at? It doesn't like being exposed to water. It really doesn't like being exposed to water. And it sticks itself into the opposing cell membrane. Right? Then the magic occurs. It changes its shape, and it draws that viral membrane right next to the cell membrane, and these things fuse. It takes them probably 20 to 30 of these hemagglutinin molecules to get a fusion event to happen. But once it fuses, everything that was on the inside of the cell, inside of the virus, now gets released into the cell, and that's when it can start replicating, because it needs the stuff inside the cell to make more copies of itself. Right? Like I said, it's taking advantage of what the cell normally does in order to infect the cell and eventually kill the cell. So, now you have these RNA molecules. They start producing sense strands, right? You have ribosomes that are going to read those sense strands and translate it into protein. You have the RNA-dependent RNA polymerase that'll bind to the sense strand and make the negative sense strand so that you can make more virus particles. The thing is, This is happening in the nucleus. This is happening in the cytoplasm outside the nucleus, so they don't actually conflict with one another. You make viral genomes here. You make the viral proteins here. They go to where they're supposed to, onto the membranes of the cell. And now you're ready to go and assemble your virus particles. Right? And so it's easy to assemble. Right? You take the cell membrane, use some of these viral proteins to create basically a coat on the inside of the membrane that attracts all of the needed enzymes and all of the needed RNAs, and then you have cellular enzymes or cellular proteins. They're going to basically pinch these things off. You've always heard of budding yeasts. Well, that's exactly what happens. These viral membranes get surrounded, right? Closes off, and then gets pinched off to form its own virus particle. It takes part of the cell membrane with it. That's how you get your enveloped virus. And then it can go off and infect other cells only after it can be removed from the cell surface. These hemagglutinin molecules bind to sialic acids. They stick there. These viral particles will stay there until neuraminidase can come in and cleave sialic acid off the surface of the cell, and then this viral particle can go away. Right? If you can block that process, theoretically you have something that can actually be used to treat the flu. So basically what scientists did is they took the structure of neuraminases. Right? This is the structure of the neuraminidase molecule. Right? This is a protein that's made up of 4 subunits, and I'm going to change the coloring here a little bit just to show you one particular thing. Again, these aren't really critical except for the idea that structure determines function. And that's really what biochemistry is. The structure of the molecule is dependent on its function. Right in here, you can see the structure of sialic acid. There's a particular location on neuraminidase which binds to sialic acid and is used to cleave it. So what scientists did is they figured out the structure of all the atoms in this protein molecule, figured out how sialic acid binds in there, and then said, okay, can we make a molecule that looks like sialic acid but can't get cleaved? Right? How many of you have ever heard of Relenza? Right? It's one of the anti-flu drugs. Tamiflu, another one of the anti-flu drugs. Basically, these molecules were engineered to fit into that little site on the protein and block its release from the cell, making it harder to produce more virus. Right? And again, they understand these molecules in, I would call it excruciating detail, but that's probably a slight underestimate. Right? Here's the structure of sialic acid. Right? Here's the structure of the oseltamivir, Tamiflu. Looks very similar, right? It's got these red molecules here, these red and blues down here. Notice how it's red up here and gray here. That's the difference between Tamiflu and the sialic acid. This molecule can get cleaved off by the enzyme. This one can't. It'll fit in the same site, it'll interact with the same amino acids, but because it can't get cleaved, it sits there and blocks the function. And this is the way scientists will actually design drugs to target particular diseases. Right? Viruses don't like to be targeted. Right? Anything that will change the way neuraminidase binds to the target molecule can basically block a drug's function. Right? Change one amino acid, in this case a histidine to a tyrosine, this glutamate doesn't bend up. See how this is sort of differently oriented? This little stretch that comes out here prevents this rotation from happening, inhibits the binding of Tamiflu. Tamiflu doesn't work on this. This is a particular mutation that basically makes the influenza virus resistant to Tamiflu. One little amino acid, and RNA molecules change really easily. Right? Tamiflu came out, what, 10, 15 years ago? Right? The virus has changed enough so that you can actually find these mutations fairly frequently. Right? This goes back to Charles Darwin and the survival of the fittest. Give it a new environment, right? Add this oseltamivir around, this drug. The virus changes so that the drug doesn't affect it anymore. Okay, switching gears away from all those tiny little molecules, something more people can deal with. Math. Everybody loves math, right? Okay, not really. This is a very simple math. Right? So, influenza, in most diseases, what you typically have is a disease starting out with a very small group of people, going up and up and up and up, peaking somewhere, and then coming down. Well, why is that? Why does this fall off? And it's the same with lots and lots of different diseases. They get bad, they start out not too bad, they expand, they get worse, lots more people get infected, and then the number of people starts going down. Right? Well, it's dependent on two major things. One is called R naught. All right? This is a replication constant, basically. In other words, what do I mean by that? Certain viruses are just more efficient at replicating. Right? Measles, recent outbreak in Texas, very, very infectious. Right? Same thing with chickenpox, mumps, COVID-19 is right up there, right? Rubella. The bigger this number, the higher the R naught, the harder it is to bring this virus under control. That's why it's so important for certain diseases to really prevent the outbreaks. Okay? But the thing about R naught is that this is an estimated number. Right? It's based on a theoretical naive cell, something that has no resistance whatsoever. Well, we all have some resistance to molecules. You can always have environmental differences that are going to help prevent the spread of a disease. And so that number has to get modified by what's known as the susceptibility. So the effective replication is basically the base replication minus the susceptibility factor, right? And so what does that mean? Well, if the effective replication constant is greater than 1, in other words, if a single virus infection can give rise to more than one infected individual, you get more people infected. The bigger the difference, the faster the disease spreads. If you can shrink RE, or the effective replication constant, down below 1, now the number of infections starts going down. You can eventually eliminate it, for now, in the population. Right? And so that's the idea. If you can increase the resistance in the population, however you do it, to a particular disease, fewer people get infected, the virus is eventually going to die out. So, we just changed the idea. How are you going to block the spread of an airborne respiratory virus? Well, we all know the answer to these, right? Vaccination is probably the biggest one for the flu. They can spend an awful lot of money, an awful lot of time figuring out how to design a vaccine for the flu. Now, there are several hundred kinds of flu viruses, and they all differ a little bit in those hemagglutinins and neuraminases. And so basically what scientists do is look around the world, see what strains are increasing versus what strains are decreasing, and they'll pick like 3 or 4 of them. Right? This is a science known as epidemiology, and they're very highly paid statisticians who do this. They'll make these predictions, and then they'll design, then they'll use those strains to make the vaccine. It takes a long time, right? And so basically, they have to track what diseases are common and where they are. If you cut all the funding from the World Health Organization, you're not going to have information from around the world. Makes it awfully hard to predict what the next strains are going to be. Right? Some of you may have remembered this old phrase in computer science, garbage in, garbage out, ego. Right? If you have bad data, you're going to make bad predictions. You can't help it. Right? You will not get the right answer. And that's the problem. If you don't know what strains are spreading, where they are, guess what? You're not going to be able to make very good predictions, and you're not going to be able to make a very effective vaccine. Depending on how accurate their predictions are, the vaccine effectiveness goes from anywhere down from 19%, which is pretty poor for a vaccine, up to 60%, which is actually reasonably good by flu standards. For the COVID-19 vaccine, they were talking 96, 97%, to give you an idea of how effective that particular vaccine was. Right? And so, basically, once you know what strains you want to make this vaccine to, now you have to make lots and lots of virus and kill it. Well, one of the best ways of doing that is to use eggs. They use about 600 million eggs just to make vaccines every year. Right? And they start this process like 18 months before they're going to put this vaccine out. So this is a long-term project. Right? But it's not all the same vaccine. Most people just get this regular flu jab, right? Pretty much all of you probably had this vaccine at some point, know people who've had it, whatever, right? Well, they make a number of different forms. Some people, older people age 65 or over, right? Our immune systems start going downhill. Right? And so you need to make a stronger demand upon it in order to get a decent response. One way you can do that is to use 4 times more virus, 4 times more dead virus in there, stronger reaction. Another way is you can use what's known as an adjuvant. Again, that one, they don't use it normally because it can have some other side effects, but the idea is to provoke more of an immune response so that you're actually protected rather than getting a flu jab and then getting the disease anyway because your immune system didn't do a good job. Another one they do is use mammalian cells instead of eggs to produce the virus. A lot more expensive, right? You have to grow these cells, you have to infect the cells, otherwise you purify it the same way. But, eggs are cheap. Well, sorry, they used to be cheap, right? Now, they're still just cheap relative to culturing cells, right? But for people who have an egg intolerance, who really can't deal with any of those other proteins that get purified with it, this is the only way of doing it. Other people just can't get injected very efficiently. Either they hate the idea, sometimes they have reactions to them, right? So they actually have developed a nasal spray flu vaccine. It's common for people age 2 to 49, so very young people can get this flu vaccine. They don't want older people to get it, right? So because it's actually somewhat more dangerous. And they use a very interesting technique for blocking its reproduction, right? This is an attenuated strand, a weakened strand, but it starts out live. Okay? And the way they weaken it is by basically making a temperature-sensitive mutation in the virus so that at body temperature, it can't replicate. It doesn't grow, so you can't get the disease very effectively. It takes a very, very unusual mutation in that one cell in order to get around this. Right? So this is a very common technique for those who can't take injections. Right? Near as anyone can tell, this is probably the best way of preventing the flu spread. If the vaccine is a good one, this will block it very well. Right? Another one, hand washing. Anybody here not wash their hands? Right? Since COVID, people have gotten more used to this idea, right? These days, you can't walk into a buffet or a restaurant without having some kind of hand wash or alcohol desanitizer around for you to use your hands. Well, soap and water is actually more effective than these alcohol wipes. Right? The reason is the alcohol is real, and the soap and water are both really good at getting rid of that membrane around the influenza virus. Believe it or not, the membrane is actually relatively fragile. These virus particles can only stand on surfaces for about 6 hours. Okay? Other viruses have a protein coat. You might have heard of a virus called norovirus, that gastrointestinal one. It's one of these protein-coated viruses. It doesn't get neutralized with that alcohol-based sanitizer. This coating is a lot tougher and can last about 2 weeks on a surface. Right? 6 hours to 2 weeks. You see why hand washing is going to be much more effective, right? So all of those alcohol wash stations inside the restaurant's good, not as good as actually washing your hands.
What's the ingredient in soap that is so...
What's the ingredient in soap, right? Basically, a soap membrane is made up of this oily substance. Ever have when people used to do their own dishes, right? You had this greasy coat on the dishwater. You put a little dish soap in, all of a sudden all that grease runs straight away from that soap, right? It's basically a molecule that can bind to this oils and just grab onto them and pull them apart. That's what it does to the membrane. And it's actually strong enough to grab onto these proteins and pull those apart as well. Right? And if you really want, I can tell you more details about that, but people don't want that much biochemistry. Right? But basically, you can just think of it as soap being a stronger way of pulling these virus particles apart, about separating them so that the RNA spills out into the air, dries up, and becomes useless. Cool. All right. How else? Mask wearing. Right? These respiratory droplets. How are you going to stop spreading them? Wear a mask. Separate yourself. Block this coughing and sneezing. I'm going to tell you a little secret. Masks don't protect you from aerosols. Yet masks are still one of the best ways of blocking, of reducing the effective replication of the virus. Why? It's actually the person wearing the mask that needs to get blocked. Whenever you go to a doctor, the doctors are always the one wearing the mask. You're the one that's sick. The goal is to protect the doctor from infecting your wounds, yourself. Right? They didn't tell us that during the COVID pandemic because they were desperate for anything to get people to stop breathing on each other. Right? So what's next for the flu? Well, the flu's going to behave just like it always has. It hasn't changed, right? February 2nd, there's this actress in Taiwan named Barbara Sue. She died from flu, age 48. Still kills people. Kills rich and famous people just like everybody else. February 4th, right? CNN talked about a second peak of influenza. What does that mean? Remember how we were talking about the idea that the replication of the virus goes up, peaks, and then drops? Well, this year was a little different. It peaked, started dropping, and then had a second peak. It became more common to be infected by this. Why? We don't have an answer to that. The fact that it happened is going to basically mean the virus is going to spread more. February 6th, CDC reported that they found some cows infected with a second type of influenza. So they had one type that came from birds to cattle. Well, they found a second type. That's not good. That means that this virus had two different ways of getting into cattle. If it can get into one other species, it's more likely to get into a third species. Maybe those pesky humans who work with cattle and birds. Right? Flu virus hasn't changed. It's with us. It's likely to be with us. And it's just something that you have to live with and try to get around as much as possible. Right? Now, there are some promising hopes. Right? If you can block the function of hemagglutinin so that it can't let the virus out of the digestive organelle, that'd be really effective. We just don't know how to do that quite yet. And there's a lot of money being spent on trying to find this universal vaccine against the flu. Right? Better hygiene. Right? There's a lot more hand sanitizer around. People are a lot more aware of washing their hands, isolating when they're sick. Not that long ago, people used to have a cold or flu, and they'd go into the office and try to work through it. That's not encouraged anymore, right? People don't like the idea of spreading all these respiratory illnesses around. It's changing, but it's only changing so fast, and we still have 20,000 people a year dying from the flu. So, it's, again, one of those glass half full things. We know how to deal with it, but we don't want to pay the price for actually reducing the spread of the flu. So anyway, that's basically what I had to talk about. Be happy to take any more questions that you guys have. All right. Sorry, Ellie seems really interested.
Why are some vaccines, like the mumps, you just get one, and some vaccines you need over and over and over, like COVID?
Okay, so why do you have some vaccines like mumps, where you only need one infection? Actually, you do need the occasional booster for the MMR. Not as frequently, but it does happen, right? And then the COVID one that they recommend every year, right? COVID is a relatively new disease. It changes still very frequently. It's one of those RNA-based viruses, just like influenza, and it's still mutating. There's a new strain called Omicron that's actually isn't blocked by previous immunities the way the other ones are. It's still better than nothing, right? So COVID, even though people haven't necessarily been infected by Omicron, there's still some resistance to it, right? But this new strain, it isn't as effective. So that's one reason. If the strain of virus changes quickly, COVID, influenza, a couple of the other ones, you need this constant booster. The other thing is that the more times your body sees an illness, the stronger the response, right? And so if you have the occasional booster, like tetanus, you're supposed to get every 10 years, and there's various lengths for different vaccines in that. The idea is that your body gets reminded of what it can make, it's primed to make it, and then it can go back into that quiescent state, but it's been primed recently, so when it does happen, you'll get more of it. So those are two of probably the biggest reasons why you need a booster for certain things. Yeah?
What is it that actually kills people? It's not the virus.
Usually it's one of those the question was, what is it that actually kills people, right? Influenza by itself, it's a sort of unpleasant, but it doesn't really kill people until you start getting things like pneumonia or things like that, or secondary infections that come along with it. Your body just gets so weak trying to fight this thing off. And like I said, you've all had this. You've had one of those illnesses that hangs around for weeks or months. All the time, you're just feeling worse and worse. And it's these secondary infections that just become more frequent. Now, the viral pneumonia is another story. That you just can't breathe well. And so usually it'll combine very badly with any other health problems that you've had. The actress I was telling you about, the Taiwanese actress, she was apparently regularly sick. She didn't have a very strong immune system, and she eventually died because she had the flu. Again, I don't know anything about the actress. This is just what I was reading in CNN when I was getting ready with this talk. Right. Other questions?
Over here.
Sorry.
When I was a kid, which mentally was ways back, soap was lye and animal fat. I don't know what it is today. Are they all equally effective because they basically break down the oils?
So the question is, soap used to be made from lye and animal fat. Essentially what it's made from now is lye and animal fat. But they don't tell you that. They'll use something different than lye, right? But the same idea is going to do exactly the same thing. All the soaps have, okay, I'm going to get into more biochemistry that I really wanted, but basically you have a big hydrophobic section and a hydrophilic section, right? So hydrophilic binds water, hydrophobic binds oil. You get enough of those things together, it's basically going to pull that oily stuff like a cell membrane or grease from your dishes. It's going to pull it apart, separate it into a little tiny itty bitty droplet that's got all of these charges around it, and now it gets soluble in water, so you can wash it down the drain rather than having it clog up your drain. Right? It's essentially the same stuff, and there's stronger and weaker detergents, but they all work the same way. And pretty much any soap is going to be killing 99.999% of bacteria, or whatever number they're throwing out this week. Right? They're all pretty much the same in effectiveness that way. More questions? All right. So I think Kit has something to add to the next speakers.
Thank you very much, Frank. As I expected, Frank had lots to share with us. Thank you. We have another presentation tomorrow. Paul White is going to talk about opioids and especially what it does to older people. And I think it's going to be a fascinating talk. So here at 8:30. See you then.
Thank you.